Okay, I think actually we can just get started. Thanks very much, everybody, for coming. This is the Northwest Science Writers Association discussion on how to communicate about health and healthcare data and statistics. I'm Chris Tachibana. I'm the treasurer of the Northwest Science Writers Association, or NSWA. Also here is NSWA Secretary Mara Grumbaum, who will watch for your questions in the Q&A box or chat, and also can help with technical issues. We're going to start with some announcements. We were hoping to get our NSWA president, Ellen Kawana, but that's the technical issue. So I will just do these announcements. Mara and I are in the Seattle area where NSWA is based. We acknowledge that we are on the traditional land of Coast Salish peoples, specifically the first people of Seattle, the Duwamish people, who are still here. We honor with gratitude the land itself and the Duwamish tribe. A little about the Northwest Science Writers Association, or NSWA. We are more than 200 science communicators in Washington, Oregon, Idaho, British Columbia, the whole region. We offer a mentorship program, career development awards, networking events, writing awards, and job postings. Each month, we highlight the published writing of our member. Coming up this year, we're planning skill sharing events, author talks, trivia nights, in-person networking, outdoors over drinks, and possibly a field trip. And we welcome members from anywhere. You can learn about our regular student and hardship membership rates at our website, nwscience.org. Okay, so today we're welcoming two experts, Doctors Rebecca Hubbard from Penn and Lindsey Leininger from Dartmouth, and they're here to help us become better communicators about data and numbers and statistics about health and healthcare. We'll spend the hour asking them questions that were suggested ahead of time, or please put your questions in the Q&A box or the chat. Our guests are joining us this evening in East Coast time, so we really appreciate that, and we will end on time. So a little bit about our experts. Rebecca Hubbard is a professor of biostatistics, Perelman School of Medicine, University of Pennsylvania. She has a PhD in biostatistics from the University of Washington, but she is from Pennsylvania, and her BS, her Bachelor of Science degree, is in ecology and evolution from the University of Pittsburgh. Dr. Hubbard specializes in developing methods for working with data from electronic health records. She was a faculty level investigator at what is now Kaiser Permanente Washington Health Research Institute in Seattle before breaking our hearts and moving back to Pennsylvania in 2014. There's a great story about how she became a data scientist. So we'll put that link in the chat along with some other bio information about her. Oh, we have our first question. What is the cat's name, Rebecca?
So my cat Perry is here with us this evening. Right now she's sitting on my lap. Hopefully she will be good and I won't have to banish her from the room.
Oh, no, cats are like super welcome. Pets, cats, we really want, we are all desperate for.
She can be very distracting.
All right, our other expert, Lindsey Leininger, is a clinical professor of business administration at the Dartmouth University Tuck School of Business. Her PhD is from the University of Chicago Harris School of Public Policy. She's a public health scientist with expertise in data-driven health policy and a passion for helping people make intelligent use of medical and public health data. As of July, she will be faculty director of the school's Center for Healthcare. She's a co-founder and was inaugural CEO of Dear Pandemic, which is an incredible resource for clear evidence-based information about COVID-19 and public health. And we'll put links for all of this in the chat. Let's see, I've got all these links, so I'll drop those into the chat. We'll start with some questions that were submitted in advance. Please submit your questions as we go. When I was thinking about what these questions, or what the answers to the questions might be, I thought all the answers might be, it depends on the audience. So we'll just sort of start from a baseline of a member of the public who's interested enough to engage in a really data heavy story. You can, Rebecca and Lindsey, please, expand to other audiences if you want in your answers. And if anyone has questions about a different audience, please let us know. So, Lindsey, I hope it's okay we start with you. Dear Pandemic monitors news and stories about health data. So what are the biggest problems that you've seen in news reports and stories about health data?
Thanks, Chris. Thanks for having me. It's so nice to be with a group of science writers and to share this panel, if you will, with Rebecca. We actually have a bunch of nerdy girls on our Dear Pandemic team who are at Penn. And indeed, I would say Penn is our stronghold. We have 4 University of Pennsylvania scientists on our nerdy girl team at Dear Pandemic. So it's great to be here. And I just want to start by saying I have so much admiration for science writers. I am not trained as a science writer. I became one by trial by fire over the pandemic. And I'm just so grateful for those of you who do this professionally. I think your work is so important in the world. And I think y'all are heroes. I do. And I say that sincerely. To get back to your question, I think the biggest issue that we have found is just a lack of basic scientific and data literacy among lay audiences. I think in our country, a lot of people don't have access to good education in data and science. I think we don't have a foundation. And I think when crisis hits, that lack of basic foundation makes people very anxious. So you have a lack of sort of basic mental models for lay audiences, and then you have fear and anxiety that hit with a crisis. And those two things I've found are very combustible and make us all vulnerable to a scary sounding headline.
Yeah, thank you. Rebecca, especially about correcting errors about where health numbers come from, like data sources and methods. What are some common problems that you see?
Well, to start off with, I would like to just echo what Lindsey said about how important communication about science, about data, about statistics really is. And I feel like, one of the reasons that I got into biostatistics was because I did see this disconnect between the language that the scientists were speaking and the language that the statisticians were speaking. And one of the things I love about my job and about biostatistics is trying to make this translation between what you see in the quantitative evidence and the scientific question that you're trying to answer. So that's something that I think about all the time. And then particularly with the pandemic, it's become really, really clear how important it is to go the next step. How do you explain that? How do you translate these findings to people who don't understand the statistics, don't understand the science that you translated it into? And it's such an important job and really, really hard, and one that I'm ill qualified to advise on. But at least I can say a little bit about the statistics piece and trying to connect that to the scientific question. And so specifically with respect to the question that you asked, there are a couple things that I've seen, especially in the pandemic, and since people became rapidly really interested in using electronic health records and big national registry data to try to answer these really important questions about health. And so even people who are very experienced scientists were making all kinds of errors in interpretation of results and of data. And I would say the two things that I see a lot of are firstly, conflating the size of the data set with the quality of the evidence. So, there are data sets that consist of the entire population of a country. We have a national registry for all healthcare encounters in our country. Therefore, we assume that we can learn from this about COVID vaccine effectiveness, for instance. That's not true. You can still make terrible errors in interpretation, the conclusions that you're making, even when your data set includes millions of people. And in fact, it's really easy to make errors in that setting because you can generate really precise estimates. You can be really, really certain about your wrong answer. So that's one thing. And the other thing, the one that probably vexes me the most, and I don't know how to fix this problem, but I definitely see this problem, is so much binary thinking, so much this works or it doesn't work. Masks are effective or they're not. We should do this or we shouldn't do this. It's useless or it's not useless. And that is almost never true. Almost everything has some sort of degree of effectiveness to it. So trying to communicate that and trying to help people to understand that, there's a gray area between completely useless and 100% effective. That it seems more important now than ever.
Oh, those are all really great points. Actually, if there is anything in the follow-up right now, I kind of wanted to ask about the education and the literacy and just sort of understanding the nuances about numbers. Is I feel like a lot of people, if they're confronted with data and numbers, just say, I'm not good at numbers and kind of give up. So how do we sort of make people more comfortable with thinking about numbers and like data and all the sort of choices that we make about them when we work with them? Any advice about that?
I can take this from my perspective. So I'm going to talk about two audiences, and one is the Dear Pandemic audience, which is 200,000 readers across social media, mostly women. But the audience that I've served almost the entirety of my career are policymakers. So I have been talking about data and numbers to policymakers my whole career. And I will tell you, the thing that gets people past math anxiety the best is a trusted, regular person. It takes a person. So I trust Rebecca. I know what she's talking about. She's there. She's consistent. She reports out. I know she's going to give me the bottom line. She's going to help me understand it or contextualize, to use kind of a more $1 million world, where she's going to keep showing up for me. And in our case on Dear Pandemic, every single number we present, we give with some sort of what can I do with this, right? This concept of what's called self-efficacy. Messaging goes down better with numbers if there is a thing a person can do at the end of it. And policymakers are the same way, right? And I have had to give policymakers some really bad data, but the pill can go down more easily. We can explain super statistically tough concepts if there's trust there, if there's two-way communication, and if I give them a thing to do to act on that piece of data. So that's what I found from my seat. Data goes better with a personal connection.
Yeah, I think that's really true about like just sort of hearing from trusted people and trusted experts and giving people some sort of action. Helping to sort of follow through on that. And actually, Rebecca, there's a question from my friend Elise, actually, that maybe you could answer. How can you talk about the potential for scientists to make errors without folks wanting to toss out the other research?
Yeah, that's very related to one of the other things that I was thinking is one of the big challenges, which is helping the public at large to understand that science is a process, not an end goal. So you're not staking your entire scientific reputation on this one answer, which is the truth. And then when that's proven not to be true, it turns out you were wrong and you're disgraced and discredited and so on. And I think that goes again to kind of the binary thinking thing, that rarely is a scientific answer completely right or completely wrong. it's sort of our best evidence at a moment in time with some amount of precision around that estimate. And, helping people to understand that, I think, is really challenging. And I feel like this is why people hate statisticians, because we so often are like, it depends, or, I can't say for sure, but, it's that kind of thing. And I do feel like with the pandemic, there is a sense of kind of breach of trust with the public, where they're like, well, but at the beginning of the pandemic, you said masks don't work, and now you're saying they do work, and and so on. And there's this real struggle to communicate to people, like, no, the evidence base has grown and changed. And even before the pandemic, like one of the specific subject areas that I teach my students about, and I kind of pick on, is nutritional epidemiology, because that's a field where you see that a lot. You see a lot of kind of evolving of the evidence and changing answers, like, have a glass of wine every day, don't, any amount of alcohol is harmful, eat chocolate, don't eat chocolate, etc. And yeah, so I also feel this also goes to your question about math and helping people kind of overcome their math anxiety. We kind of have to go back early in the educational trajectory and start there with helping people to understand that science is a process, and math is not, being good at math is not a trait that you're born with and you either have it or you don't have it. It's a state that you enter into because you practice it a whole lot and you use it. And there are very few other fields that we treat in that kind of dichotomous way. You're either good at it or not. But math, we really do. We convince people you're good at math or you're not good at math. And then the ones who aren't kind of throw up their hands and feel very anxious about it. So yeah, that I think starts very early on, that indoctrination into the concept of like, you're a math person or you're not a math person. So that's very difficult to start to fix.
Yeah, those are really great points. So maybe as science communicators, we can just sort of think about that more, just keep saying, like, this is what we know right now, and we expect it to change. And especially, I guess the pandemic is a really great example of that.
And Chris, can I hop in
Yeah.
here on that? So I just want to amplify what Rebecca just said. And because she's a biostatistician, she might appreciate that there's a nice piece of experimental evidence in science communication that demonstrates that while our brains hate uncertainty, that's why they don't like statisticians or policy analysts or economists who are like, well, it depends. Our brains hate that. They're wired to hate that, right? But there's a nice piece of experimental evidence that says if you consistently socialize people to expect uncertainty, you can minimize the harm. And so at Dear Pandemic, to operationalize this, we write on what's called the information beat. So we write consistently on how to think like a scientist, how to be okay with uncertainty, how to make peace with changing data. And I think that as science writers, we all can help our audience's brains be more okay with uncertainty by socializing it being okay.
Yeah, I know that actually, I think you had a couple of slides, if you want to share any of them about some of the Dear Pandemic quiz transparency. I think it's like one of your big principles.
That's right. That's exactly right. And we, there's a nice literature. It's called emergency risk communication, and it's how to talk about data when the world is on fire from a public health perspective. And one of the key rules is transparency. And again, socializing people to uncertainty, teaching people what science can and cannot say. And when you get it wrong, say you're wrong. So we got it wrong on masks in the beginning, like so many science communicators. We owned up to it. We got it wrong on Advil. There was a very early data signal that one should not take Advil. This was like way back in like March 2020. People probably don't remember it even. It came from the WHO. We were like, maybe don't take Advil. That was wrong. We owned it. Right. So I think it's this trust over time, transparency, and I think everyone teaches, everyone learns in our community. I think that also helped defang the fear of uncertainty of data and sort of introduces this concept of risk as a dial as opposed to black or white thinking or binary thinking, right? Like we're all doing our best. We adopt what's called harm reduction. It's not all or nothing. The data aren't certain. You know, we're going to fly through this uncertainty together with the best thing we have, which is a scientific method. And we will be here for you, reporting out what we can when we know it.
Yeah.
Which is
I
what you
hope
will
that's our
do as
goal.
science writers. So thank you.
I hope
I
that's
mean-
our goal. I hope that's what we're communicating. And so we are coming to another group of questions that Diane in the question and answer has very neatly expressed. And that is the click crazy media style doesn't help with this stuff. Ideas for relaying nuance to the public. Rebecca, we have a specific example, but do you want to give sort of a general answer before I throw the specific example at you?
Let me give a general answer, but I don't think that it's a good one per se. I would just sort of, because I think it's incredibly difficult. You know, it's difficult to convey nuance in a whole article. Conveying nuance in a headline that a lot of people aren't even going to click on seems like an extraordinary challenge. But you know, I would just kind of come back to this idea of making sure that we incorporate into the information that we're putting out there, even if it's, you know, in the headline, the concept of the uncertainty in what we're saying. So, you know, it's not if vaccine efficacy is 55% or 60%, that's not actually as important as how certain we are about that. If it could be anywhere from 40 to 70, that's the more important piece of information to communicate than that our very best guess is 51, 52, et cetera. So, you know, if there's a way to kind of emphasize that, how precisely are we able to say this, as opposed to just putting all of our eggs in the basket of the one estimate. That's what I would try to do, but how you do that in the one thing somebody's going to see before they keep scrolling, I'm not sure.
Yeah, but a range, like, you know, sometimes giving a range is a little bit helpful because we're kind of used to that. And so I am going to come to the question that we got. Oh, go ahead.
I don't break, but someone who did really well with this was Angela Merkel.
Oh.
So when we talk about a range of estimates, if y'all remember, Dr. Birx at the very beginning of the pandemic got up at the news conferences and was like, here's what our infectious disease models show. We could have anywhere from 0 deaths to 10 million deaths. I mean, it was just like, what? I don't know what to do with that. But Chancellor Merkel was like, if this virus is the worst of our estimates, we overrun our hospital capacity by June. If it's the best of what our infectious disease modelers are saying, we overrun our hospitals in October. So, you know, she benchmarked the uncertainty in just the way Rebecca was talking about. Like, she actually benchmarked it. So she took that 40 to 75% efficacy number and actually, you know, and used the benchmark as opposed to just threw out a number or just said there's this huge interval, we're so uncertain. I'm not doing a good job of explaining this. I'll find the write-up of it and drop it in the chat when y'all are answering the next question.
But that is such a great point, is that maybe a range is not so helpful, but the consequences of it, like explaining that, it makes it a little bit easier for people to imagine and gets them a little bit closer to, like, what could I do about it? You know, I could do something to try to stop our hospitals from being overrun. That is helpful. I did want to get to this because someone specifically asked this on Twitter, and it was a comment about AUC of ROC curves as a quote unquote pain point. They actually use that term for the area under the curve of receiver operating characteristic curves. They're often used to kind of graph out accuracy related features of a test, like a cancer screening test, which is something that I know Rebecca works on a lot. Rebecca, I'm going to show this, and how would you explain this?
Okay, so here we are looking at some ROC curves for some different biomarkers. So ROC curve represents like the trade-off between identifying true cases and accidentally mislabeling people who are not cases as being cases. So that's what we have on the top. And then on the bottom, we have kind of a binary, like a confusion matrix version of this. We're looking at true positives, false positives, and so on. So ROC, you see these all the time, and I think they're only going to become more common because we're developing more and more risk prediction models, machine learning, AI, all of these sorts of things that do give you like a risk score. And the ROC curve on the top here is one of the ways that people look at that and say, you know, here's how we evaluate the performance of this thing. So I do see this becoming more and more common. The thing that's terrible about the ROC curve is it's looking at this trade-off between different kinds of misclassifications across the whole range of your biomarker or your risk score or whatever. And it doesn't tell you anything about the harms or the costs of the different kinds of misclassifications that you might be making. So, you know, if I were trying to present this to a general audience, a non-technical audience who had never seen an ROC curve before, I would probably not do that. I would probably pick a threshold in my biomarker or in my risk prediction score that results in something that's meaningful. So for instance, if I can only afford to run so many follow-up examinations on the people who I flag as being high risk, that's going to inform where I need to cut that biomarker. And so at that cut point, then I could say, all right, here with this risk score, here's the probability that we're going to identify you as a high risk person if you actually have the disease, and here's the probability you're going to be falsely labeled as such if you don't. Actually had a situation recently with a collaborator in oncology who had developed a tool for identifying people who were at high risk of experiencing cancer treatment related toxicities. And they were going to use this tool to try to triage them to whether or not they needed to come in for an office visit to evaluate them for experiencing an adverse event. And in that setting where what you're triaging people to is a pretty low harm, low cost intervention, you want to make sure you catch everybody who is experiencing a toxicity, but it doesn't really matter if you flag some people who aren't and they have to come in for an extra office visit. So that was actually a setting where we said, well, we don't really care much about specificity at all. We can afford to make a ton of false positive classifications. We want to make sure we catch everybody who actually is experiencing the event. So, you know, it's totally context dependent how you would want to communicate about that, depending on kind of what's the follow-up test that comes after the biomarker or the risk score. But I would, you know, it really needs to be contextualized to the specific kind of test, the specific kind of follow-up. What are the consequences of different kinds of errors? The ROC curve itself is way overused. I think people just kind of do it as a knee-jerk reaction, and then they summarize that with just the AUC, which is even less useful. So you see a lot more than I think you really should, because it's not a terribly useful tool in a lot of settings.
Oh, that's interesting. I think the person sort of like brought that up is because you see it so often. But I think like when I've had to deal with it, what you've said is correct. I mean, I can understand when someone gives me the context or the circumstances and say, you know, and they say, the consequences of missing someone is really serious, you know, or the consequences of a false positive or something, or a false negative is really, you know, that's what we want to avoid. And then you can kind of understand, like, that's why, you know, we're focusing so much on that and not so much on maybe one of the other factors. So yeah, like this science writers say this all the time, but it's all about like storytelling. Lindsey, do you have anything to add about nuance or this specific example?
Well, you know, it's so tough because the C statistic as a researcher, I have to hit a C statistic of 0.7 or above in certain prediction contexts, or else my paper has no chance of getting published, right? And that's unfortunate. It's almost like the p-value of predictive modeling, right?
Mhm.
So that's tough, and I don't like that norm. So I also kind of fight the I fight the good fight and say, let's talk about settings where a false positive is more costly versus a false negative. And I work in a lot of low resource settings where a false positive actually is quite costly when
Yeah.
you're resource constrained.
Mhm.
So often I'm much more worried about positive predictive value, which is, am I actually flagging someone who really is going to need these resources because I don't have so many resources to spend? All of which to say is I like contextualizing this in the cost of a false positive versus a false negative. And I always teach my students that it is a management or clinical problem where you draw that threshold, not a math problem.
Yeah, that's a good point. Also to think about, what's important to the people you're talking to or writing to or something like that. Is it cost, or is it avoiding a certain diagnosis or something like that? Yeah. Well, we had another question that was sort of related to a nuanced topic about significance and trends. Lindsey, maybe you could take this one. I'm just going to read it because it was so good. So someone wrote, "It's very common that a researcher I interview for a story tells me that while their clinical trial's primary outcome measure, let's say overall survival, wasn't, quote, statistically different between fancy new experimental drug X and standard of care, there was a, quote, positive trend suggesting a possible benefit of fancy drug." So the writer's question was, this is total BS, right? But as a good science writer, the person wrote out that BS abbreviation in full for clarity. Like, I don't know if I can say this, but yeah, Lindsey, how do you communicate about like trends and significance and stuff like that?
That's tough. I mean, so again, this is where the context matters. But when I've had to talk about results where, sometimes I will look at something that is not statistically significant, but it moves or informs my priors, right? So I'm not willing to throw out something just because it doesn't meet traditional levels of statistical significance. You report all the facts. So this is a large magnitude. This indicates a large clinical benefit, but this difference does not meet statistically significant thresholds, right? Like I think you have to lay it all out, because the one thing I've learned about reporting on risk, reporting on margins of error, is that risk lands differently for different people. And some people care much, I'm not going to not take a drug because the P is 0.06 instead of 0.04. That's not what's going to do it for me, right? So I think you have to just be really honest about just the facts and a little bit of context. But that's my own personal.
Yeah. Okay. So you would allow someone who said it's not significant, but I see a positive trend.
No, I would report the effect size. I would report what the p-value was in plain language. And I would say perhaps that does not meet traditional standards of statistical significance, which are blank.
Yeah.
But we don't want to put all of our eggs in the traditional standards of statistical significance either. So yeah.
Oh, yeah. And I see.
But I don't like the trending towards significance. I don't do that. I don't know about you, Rebecca, but I don't do that.
I know trending towards significance is so bad, and I think they like take away your statistician card if you use that language. I think I actually contractually have to be opposed to trending towards significance. I hate trending towards significance. In my own science, I agree very much with what Lindsey just said. You did the study, you get your estimate, it's got a confidence interval, it's got a p-value, you report it. It doesn't matter if it was 0.07 or 0.04 or 0.5, whatever, you report it. and, let the reader, hopefully a reader who understands what any of those things mean, interpret it. And that's always been sort of my approach. However, it is a little bit different in the context of confirmatory clinical trials. Trials are being used for regulatory decision making versus pretty much everything else. Because if I'm doing a study outside of that setting, and individual people or individual scientists are going to read it and decide, okay, do I think that there was a signal there or not? Am I willing to go with that P was 0.06 and I think that's good enough, and do future science building on this result because I trust that, or I think that's worthy of further investigation? That's a different setting than the FDA has to make a decision. Are we going to approve this drug and allow potentially millions of people to be exposed to it, yes or no? And so I do really agree with, when you're in the setting of a confirmatory clinical trial, you decide up front what is the type 1 error that we're willing to accept, and then you got to live with that. Whatever was the decision that was made when you pre-specified the trial, that's the basket you're going to put your eggs in. And if the p-value is 0.051 and you said 0.05 was the threshold, like, you lost. that didn't do it. It didn't make it. It's unfortunate. And that really is one of the only settings where I am kind of like, no, you have to make a binary decision, because the FDA has to make a binary decision. The regulatory decision has to be made on the basis of some criterion that we understand the properties of that criterion. And so that's the way we've decided to do it. And we have to live with it. Once we decide we're doing it that way, we have to live with it. But in broader settings, outside of that very specialized case, I completely agree with Lindsey. Like, put all the information out there and let people interpret it.
Okay. Yeah, actually, this is a good tip also, maybe for science writers who are writing in this field, is for something like that, ask, what were your pre-specified outcomes? Like, before you started, did you write down what you would say is like significant, not significant, meaningful, not meaningful, or something? And then how did that stack up? That's a really good, there's something of a comment, like I think effect size came up. Is that something that's meaningful to people? Is that intuitively easy for the public to understand?
In my experience, no. And I was told my first year of epidemiology, you are responsible for interpreting your own results. So I think it is very important that researchers, or in my case, a science writer for the public, doesn't say this has a 0.2 standard deviation effect size, right? Again, here again is where I really think it's incumbent upon us to do the Angela Merkel and benchmark it to something that is meaningful, a meaningful metric.
Yeah.
I think that's really important. So yes, I believe in effect sizes. I believe, I like all the numbers. I like, you present it as an absolute risk difference. You present it as a relative risk difference. You benchmark it to something like a clinically meaningful metric for your audience, or in my case, a policy meaningful metric for our audience.
Okay.
I think that one number, you have to add a lot of context around.
What about Rebecca? What do you think about that? Or number needed to treat sort of came up in a discussion recently, if that's something that is meaningful for people.
I also do not like standardized effect sizes. I feel like standardized effect size is what you get when someone doesn't want to invest the energy to try to think about what their estimate means in the context of the scientific questions. They're like, well, we'll just divide it by the standard deviation and tell you what the standardized effect size is. They're very, very difficult to interpret in a meaningful way. And, people kind of have these rules of thumb of like what's small, medium, large, which is completely divorced from anything to do with, with reality. It doesn't measure up in any way relative to what might be a important health effect, or, something that actually means something in a clinical scale. So I try to get people not to do them as much as possible, because they are tough to interpret. Number needed to treat is interesting. My clinical colleagues love number needed to treat. It feels meaningful to them.
Okay.
I feel like it's one of those things, sort of like hazard ratios, that they've seen so many times, they think they understand what it means, but I'm not sure. And to the general public, I'm not sure that it means what people think it means.
Oh, that's interesting. Okay, I did see some criticism, or I sort of saw people saying, this is a really good way to illustrate something. And then I saw people saying, no, not so much. So that's interesting. Let's see. We had a question. Both of you, I'm sure, have talked to writers or people who are going to tell your story, and had to figure out how to address a certain audience. So the question is, like, how do you get scientists to break it down and not use science speak? Lindsey, what do you?
Boy, that's hard. And at Dear Pandemic, we're trying. I mean, we talk about our 200,000 readers, which we're really excited about. But the thing I'm actually most excited about is that we now have over 36 women scientists who have been trained to speak science to lay audiences, right? So I'm trying to scale that side of the platform, like help scientists learn how to use plain language more, because I am not sure if I'm going to a scientific source, I'm not sure they're going to know how to to speak science to lay audiences. It's a completely different skill. So I think that's where this translator function's needed. And that's where the science writers come in. That's where people like me come in. That's where people like the now defunct, but still lovely Health News Review. They have wonderful explainers of these concepts, right? I think we have to de-science what comes from the PIs. I really am trying and investing in helping women scientists speak science to lay audiences, but I don't think our institutions are set up to. If you think about what people in academia and science are promoted to do, it's to write science speak to scientists, and we have a precision with our language that's like a shortcut. So I think that's what science writers, people like me, I think we translators kind of have to do it for them.
Okay.
And y'all excel at this. I mean, I think science, I really think this is the science writer wheelhouse. So science whispering, indeed.
I mean, I've heard the tip before is just like, as a science writer, don't act, even if you have read the paper and you feel like you understand the paper, go talk to the person like you have no idea what, it's about. Otherwise, you'll sort of get into that, like talking with the words of art that they're going to use, and you're not going to be able to use that in your piece. But I don't know, Rebecca, what advice do you have about talking to a scientist in a way that'll get answers that are understandable to a broader audience?
Well, I think what you just said about, kind of approaching the interaction as if you haven't read the paper, I think that actually is a really good idea. I had never thought about it quite like that, but I do a lot of talking to scientists about topics that I don't know anything about. They need me to help with the statistics, but they're in some field that I have absolutely no experience with. And so I do a lot of that, and, kind of listening and then repeating back to them the words that they just said to make sure I've figured out how to use those words. When you use this term, what is it that actually means, and trying to learn to speak that language is helpful. But I think that, as I do that, they sort of learn how to talk to me as well. They sort of become aware, they become sensitized to the jargon that they're using, which there's so much of it in science. There's so many levels, so many layers of jargon. But I also think, kind of along the lines that Lindsey was just saying, one of the things that I've observed in the pandemic is how many different kinds of communication there are, different audiences that we need to be communicating to. And I've seen some of my colleagues trying to use like Twitter, for instance, as a platform for disseminating results. And some people are really good at it, and some people are really bad at it. And what I would say is, I don't think everyone needs to be good at it or should be good at it, because I think that function of translating the science to another audience is its own really important skill. And expecting someone to be, a world-class scientist as well as a world-class science communicator and translator is probably unrealistic. So, the people who are able to do it it's amazing and wonderful. But I also think that translation function is super important. There's no reason, just like we sort of assume that just because you're a good scientist, you would be a good science educator. Well, that also is a ridiculous assumption. There are plenty of people who are very, very good at what they do, but can't teach someone else how to do it to save their life.
Yeah, that's a good point. That's good. We all of us science writers and editors and everybody have jobs for the future for those people who have difficulty with that particular skill. I hope. Thanks. Well, let me ask a question that had come up that's a little bit not on one of these topics, but do you have advice? I'll just open it up to either of you on reporting on numbers that show health inequities without stigmatizing or further marginalizing groups that are experiencing the inequities.
I have a little bit of thoughts about that I don't think it's I don't think it's terribly nuanced. I mean, the thing that I have been trying more to do, because I've certainly done many studies, for instance, where we include race as a covariate, say, and then we just sort of slap in a table, oh, here are the differences in outcomes by race. And so what I'm trying to become more conscious about in communicating those kinds of results is, firstly, recognizing whether or not we're in a context where race just represents racism. Does it represent the way that society interacts with someone based on society's perception of their racial identity, first of all? And secondly, making sure that if we're going to present that, if we're going to show, for instance, differences in health outcomes by race, that we also consider what might be contributing to those, because it's probably not biology. It's probably a lot of other factors that we are, that are confounding the race effect. So I think being sensitive to both of those two things are things that I'm trying to work on.
Okay. Yeah, thanks. I feel like maybe science journals are getting a little bit more careful about that. And I hope that sort of goes over into the things we write about people's work and study. Lindsey, do you have anything to add to that?
No, I just amplify that. And I'm very excited about the Mackenzie Scott recent gift to the Kaiser Family Foundation to really make some headway about collecting data on race and healthcare and thinking about how to present it and communicate it well. It was a big gift in health equity data, and it made me excited.
Okay, I actually did hear about that, but and also to KFF, to Kaiser Family Foundation, is a great source of news.
Yeah.
So that
I mean, I'm not an expert on this. I am a learner, and I think stressing that all the things that Rebecca just said are so key.
Yeah.
I mean, I think a lot of people don't understand that there is more variability in health outcomes within racial groups and across racial groups. I think people on this call know, but I think most people in my world who aren't health people, like I tell my business school colleagues that, and they're like, really? Like, we have work to do. So.
Yeah. Oh yeah, I should mention that Lindsey's class is for people who are it's future healthcare leaders or something, right? So that is kind of thanks, Mara, for Mara found the story about the Mackenzie Scott donation. Yeah. Great, thank you. Let's see. One of the other questions that's sitting here, it's actually from Ellen. It looks like it's from me, but are there good online resources you'd recommend for questions such as can an alpha value be B1 or greater? Like, yeah, what are some good resources to help us be better communicators?
I don't have a good answer to that question. I hope Lindsey has some thoughts about that.
I do. Gosh, this isn't a comprehensive list, but I like the Center for Evidence-Based Medicine out of Oxford. I think they have some nice explainers. I like Health News Review. I like that website. It actually has like how to talk about an odds ratio in a news report. My colleague here at Dartmouth, Stephen Woloshin, does a lot of training on medicine in the media, and he has fact sheets and explainers that are geared towards journalists that explain things like number needed to treat, which is a metric I actually don't like and don't find intuitive. I do think the evidence-based medicine folks at Oxford, I think Stephen and the medicine and media people have done a really nice job with their resources.
Okay. Anybody who's on the call, if you want to drop things into the chat that you know of that are good. Oh yeah, okay, thanks. Thanks, Mars, like looking everything up for us.
Thank you. I'm sorry. I'm like trying to Google and talk.
Like, what is it?
Makes for a very chaotic viewing. I'm sorry that I'm trying to do it at the same time.
Yeah, and Health News Review was a site that would, it collected scientists from all over and science writers, and I think you're right, like they have not continued because of lack of funding, but all the information they put up before is still there.
It's still there. So I just dropped one on odds ratios, for example. So I use a lot of this stuff in my teaching. So Google is a good.
Yeah.
Everyone says Googling's bad, but I'm not going to lie. It helps me a lot.
Oh yeah. I mean, if you're sort of selective about what you look at, I guess.
Right. Like, this audience is full of savvy Googlers. So
yeah, we could probably, I don't know, find things. I wanted to ask Rebecca, because I know that we got some assistance in promotion from a big network of patient research partners and patient advocates. And so I kind of wanted to ask this question for them. I know you did a project that was funded by the Patient-Centered Outcomes Research Institute, PCORI. And the requirement for that funding agency is working closely with people who will be affected by the research. And so I saw that your project had partners who were patients, parents of patients, patient advocates, as well as like medical professionals. Like, what did you learn from trying to present like really detailed research evidence to those different groups?
It was a really interesting experience. So the project that I had was a statistical methods project. It was very technical, but we had this stakeholder partner panel that was part of it. And throughout the project, we would have quarterly meetings with patients and parents and clinicians. And it was a learning process for me over the course of this project. So when we first started out, I had a very naive idea. I thought, okay, I will teach them about basic statistical concepts. And then once they're kind of up to speed on statistics, then we'll all be speaking the same language, and it'll be bi-directional communication and it will be great. No, that was not a realistic expectation. I was not going to teach them statistics through a series of several quarterly meetings over the course of a couple of years. So after I sort of divorced myself from that idea and stopped trying to be didactic and just sort of accepted that everyone had a unique contribution to make to the project, was actually really, really, it was really, really interesting. So what we would do was not try to present to them the technical details of like, here's how the statistics work and here's the programming that we did, but show them in terms of things that would make sense to patients and parents. So we were trying to use electronic health records to do early identification of kids with type 2 diabetes. So we would show them, if you apply this algorithm that we've developed, here's how many kids you identify, here's how this is different from if you'd used a different approach, here's the characteristics of these kids. And then we asked them, is that consistent with what you would expect to see based on what you experienced, like in your symptoms before you were diagnosed, or with the clinicians and the patients that you see in your clinics? And they had really interesting insights. And like, to my total dismay, we ended up doing a qualitative project. So one of the papers from the statistical methods project is actually a qualitative paper where I didn't do the interviews because I'm not a qualitative person, but the qualitative people talked to patients and parents and asked them, what did you experience as part of the diagnostic cascade related to diabetes? So yeah, it was a very interesting process of learning how to listen and how to respect everyone for kind of bringing a different piece of the puzzle to the table, and not trying to make I wasn't going to become an expert on the biology of diabetes, and they weren't going to become an expert on statistical programming, but we were all still able to contribute. And yeah, it was fun. I think PCORI actually subsequently relaxed the requirement that statistical methods projects have a stakeholder panel. So I think I had one of the few projects that did do it, but it was totally worthwhile and beneficial. It was really interesting.
Oh, that's okay. That is interesting. I thought you were going to explain to us like how to tell all these people about, but it
Well,
just like
the one thing that I think what did work slightly, and it wasn't super beneficial from the perspective of advancing the project, but I do think it was kind of effective, was I did teach the stakeholders about Bayesian statistics. And I think, I'm sure that all of them had never encountered Bayesian statistics before. And I think it did actually work. And I think Bayesian statistics actually is more intuitive to a lot of people than the traditional classical way that we teach statistics. So, so I think maybe that actually is a worthwhile thing to try out, is if people are struggling with statistics, I think Bayesian statistics actually makes more sense to a lot of people than classical statistics.
Oh my gosh. Okay, we are running out of time. Otherwise, I would have you like go through that, because that is my huge question, is I want someone to really explain it to me. But let's get to this last question. What do you both think of the Cochrane review? Lindsey, what's your feeling about Cochrane?
I like it. It's pretty narrow though, as a population researcher, right? In my world, most of the influences on your health originate and are fought outside of the clinic. So I don't really look at clinical populations. I look at populations, some of which end up in the clinic. So for policy work, it's not as, when I can use it, I love it. But for a population level researcher, it's hard because my denominator is almost always inclusive of people who aren't patients, so or aren't yet patients.
Yeah, that's true. Maybe for you too.
I'm grateful for the effort. I mean, what an amazing effort. Rebecca, you might, you're in a med school, so you might have a totally different.
Well, as a person, I agree with you. Most of life takes place outside of the clinic. Most of what affects our health takes place outside of the clinic. So I completely agree with that. As a medical researcher, I do think Cochrane Review is useful for sort of aggregating what's the best evidence that we have on any particular topic. There's a whole field of thinking that there are way too many meta-analyses and research syntheses and meta reviews right now. And there is a bit of that, but at least with Cochrane, I do feel it's sort of the gold standard in terms of quality. So I would put some faith in that.
Okay, we have an endorsement for, and there's, I saved the chat, or I'm going to save the chat. So we have the other resources. If anybody didn't get to copy them down, contact me, chris.tachibana@gmail.com. And that's the end of our time. Thanks everybody for joining us, and special thanks to Drs. Hubbard and Leininger. We'll post this recording on the event page and nwscience.org as soon as possible. Thanks very much, everyone. Good night.
Thank you.
Thank you. It was really fun chatting with you.
Thank you.